So, I mean, I think we all know what nanopore sequencing is and why it's important. So to really highlight the importance of why this is great, previously Dorado, which is the base calling software from nanopore, was NVIDIA only, but you've made it actually hardware agnostic. So it now runs on AMD and NVIDIA, and you've got a significant speed up as well, which is a great achievement. So the format we have here is do a bit of an interview first and then get into the live demo, which will be pretty exciting. So could you share the story behind Slorado's development? Like what specific challenges were you trying to address that weren't being met by existing solutions?
Yeah, sure. I'll answer that question. So how it started was we wanted to do some acceleration related research with base calling. And when we looked into Dorado, we realized that it's too complicated. So we decided to create a simplified and a streamlined version of it, which is manageable, understandable, and suitable for a research purpose. And then that's how Slorado began. And while implementing that, we found out that Dorado, even though it is open source, there is a library called KOI, which is closed source and seemingly only support NVIDIA GPUs. So we thought, okay, we would want to make Slorado fully open source by writing whatever the KOI does in an open source fashion and enable support for like a wide range of hardware. So that's how Slorado development began.
Right. Yeah, I mean, having this central thing, which is closed source and giving you this vendor lock-in is pretty difficult. So for our technical audience, could you explain the sort of key innovations that allow Slorado to run effectively on AMD, whereas previously it wasn't possible?
Yeah, I mean, like Cindy said earlier, if you want to do state-of-the-art base calling, you have to use state-of-the-art models, which is only provided by ONT, and they provide Slorado, which implements all the performance-critical optimizations behind a closed-source library, right? So really, the only thing we were trying to do was open source this optimization part behind the closed-source software and make it open source. And really, once you have like a implementation on any GPU, you can move that to whatever framework you want. So for us, it was AMD.
Nice. Yeah. Do you think there's any particular use cases that are especially well suited to this type of using Slorado?
Yeah, I can answer that question. So in nanopore genomics, base calling is the first step before doing any other analysis. So as you may know, that's the first step. So for any research related to genomic base calling with Slorado can be beneficial, especially if you have access to a lot of AMD GPUs like that we have on Pawsey. You would be able to quickly get your job run because there are so many GPUs around there. And we have seen that the queuing time is very low, that the runs instantly run there because there are many free GPUs. So especially when there's an urgency to get a large number of samples quickly base called, yeah, you can kick a lot of jobs and get it done there. Yeah.
And so could you walk us through a specific research scenario where Slorado has significantly improved workflow efficiency or maybe enabled new types of analysis?
Yeah, I think it's especially when like the urgency is important. Like let's say you have hundreds of like data sets that you want to quickly base call in a newer model or like using a particular version, then having access to this many like, free AMD GPUs, which are not like, heavily like used is very beneficial. So you can quickly get the jobs finished. And another side of Slorado is for researchers like ourselves or computer scientists like ourselves who want to accelerate base calling research. Because it's a simplified version and fully open source, people can take it and adapt to whatever the platform they need. It can be other types of like GPUs or hardware like devices like FPGA. So it allows a broad set of such future research too.
That's exciting. If you could get it working on FPGA as well, like that really opens up a whole bunch of new applications, which is really exciting.
Yeah.
And so would you say that, well, the question is, how did Pawsey's photonic supercomputer enable the development process? Was that largely to do with the availability of these AMD GPUs, or do you think there was more to it?
I mean, before we switched to Pawsey, we didn't really have access to anything as powerful as the MI250Xs they have on Photonics. So we were pretty limited in our continuous testing and verification pipeline. We're only doing like 20K reads at a time, which isn't that great of a test for robustness. So now that we switch, we can easily do like 16 million reads for all our models within like 2 days, right? So it's been like a really big speed up for us in the development process.
That's awesome. I really love to hear when there's been some kind of like success story like that. I get a bit excited, which is maybe I need to get out more or get a hobby, but that's really awesome.
Talk to the wrong people for that.
Yeah, right. Probably eat, sleep, bioinformatics, repeat, right?
Yeah.
So how easy is it to integrate Slorado with existing bioinformatics pipelines, and what should researchers expect in terms of compatibility?
So just like Dorado, it's a command line tool, a command line bioinformatics tool. So one can plug and play easily because we follow a similar, like a command line interface to Dorado, similar outputs and similar command parameters. Because of that, it should be seamless to switch another base caller with Slorado, we believe. Yeah.
Okay, yeah, that's great. And the installation is also, you've got this nice precompiled binaries. That's also quite nice. You don't even need a container.
Yeah, that's one of the like benefits you have in Slorado, even in Dorado. So here we have provided it as a like a bunch of tarbold binaries. So you have to just extract it and run it. It will run on most of the available Linux systems as we have tested. But of course, if you want to go and compile, if you enjoy compiling by any chance, we have provided the instructions for that too.
Yeah, there's always someone that does. So it's good to have the option. I was used to impress my boss compiling stuff. He would look over and see all the output on the screen. I think I was doing something really amazing when all I did was type make, install, or such computer science. Actually, you can get Slorado to scale to 8 GPUs simultaneously. So it's using a whole node on Photonics with the AMD GPUs. So could you explain the scaling benefits and when researchers should consider using this multi-GPU setup?
Yeah, so I mean, for any small data sets or anything that would run particularly fast from default parameters, you probably wouldn't consider something like a multi-GPU setup. But with the new transformer models, or if you're using super accuracy, it is quite hardware intensive. So that's when it would make sense to take advantage of that parallelization for your jobs.
Nice. And so it works on like one whole node. Does it have multi-node scaling at the moment?
So at the moment, like Slorado binary itself doesn't go and take multiple GPUs from other nodes. But what the user can do is simply like submit an array job. Like if you have a data set split into small like batches, then they could simply submit a huge array Slurm job that go and in parallel do the base calling. Because the base calling process is a very embarrassingly parallel, we call an application. like every read is independent of the other. Therefore, you can divide it across as many nodes as possible if you want. Yeah.
That's great. So you could really put photonics to the test and see how many nodes you can do all at once.
Yeah, yeah.
Something exciting to do on a Friday night. So what's your roadmap for Slorado? Are there features or improvements that you're currently working on?
Yeah, Pawsey is working on a number of them. He will comment on it.
Yeah. So we'll have that support very soon. And at the same time, we'll pick the people to implement optimization for the inference side, so the neural network side of things. So that's like a whole nother half of the base calling pipeline we haven't optimized yet that we're looking at. And then obviously, Jacinda touched on this before, but we still have time. So later on, we're thinking about looking at embedded systems or FPGAs, things like that.
Yeah, that's exciting. I'm really interested to see what more you'll do with it. And so how do you see Slorado contributing to the democratization of high performance genomics analysis globally?
Yeah, I think it's mainly by enabling the base calling process on like a number of HPC systems. So previously with Dorado, it's only limited to clusters where NVIDIA GPUs are there. But now Slorado works on both NVIDIA and AMD, which are like the two major vendors in high performance graphics card. So we believe by broadening the number of vendors, we can, make it available to a wider community. And in the future, we are also thinking of enabling the support for desktop, laptop, and smaller embedded GPUs from both NVIDIA and AMD, so that people who can have access to high performance computers, but something else can still use it for certain applications. For example, in the field applications where you can't like just carry a huge server with you unless you are going to the gym. You can like take a laptop or something, do the analysis there itself, right? So for such cases, we are planning to develop optimizations.
That's awesome. We're really enabling this kind of, everything from like a huge scale to quite local, small scale, or just being out in the field, which is something cool you can do with nanopore. And on the note of making it more, available across many different clusters, there's quite a few really large clusters around the world. For example, for our Swedish guest, the Lumi system in Finland basically has the same architecture as Pawsey, a giant photonics, or we are a mini Lumi, depending on how you want to look at it. So what you've done is actually enabling this type of research to happen around the world. So it's actually a really big deal. So actually, we have a live demo, which I hope will work. I believe in you, Jonathan. Live demos are a bit cursed, but you're clearly pretty talented. So if anyone can do it, you can. But before we move to the demo, could you just briefly outline what we'll be seeing and what aspects the audience should pay particular attention to?
So yeah, this is going to be a very gentle introduction to just the basics of the tool. It is tailored towards Pawsey and their Slurm workload manager, but it's simple enough where you can generalize it to anything you have in mind for your own workflow. If you do have to remember one thing from this is that we have a GitHub and all our information is up on GitHub, and if it's not, you can make an issue, right? Yeah, so just get started.
Yeah, let's share the screen. That's the first hurdle in a live demonstration. It seems like we have successfully nailed that.
Can you guys see that?
Yeah, looking good.
Yeah.
Okay, so I mean, the first step to actually using the tool is obviously downloading it. So you'll find that on our releases page. on our GitHub. You'll see we have two compiled binaries. If you're running on NVIDIA, you download the CUDA binary. So this is a CUDA binary package. But since we're running on AMD, on Pawsey, we'd be downloading the ROCm binary package. Since we don't want to waste any time waiting for that to download and install, I do have a prepared version here. You guys can see that clearly. Well, that's probably a good size. So this is our pre-installed package. If you were to look inside, oh no.
Okay.
I'm gonna have to SSH. All right, you guys get to see it from the beginning.
Yeah. We had a SSH connection established, but it seems to have got stuck. So we are SSHing to Setonix again.
Okay, so this is our package. If you look inside, you'll see a couple of folders. The most important ones, the only ones you have to care about is the binary one. So if you look in the binary folder, this is our Slorado binary. This is what you'd be executing. And the other one is the models folder. And that just contains all the prepackaged models that you'd be using to run basecalling. So to actually run this, we're going to allocate a interactive session. So on Setonix, we use the Slurm workload manager to allocate a free GPU node. So we'll specify that partition. We'll ask for, we'll ask for one GPU. And then
GPU will, yeah.
We'll ask for a single node and specify our account. Okay, so this is going to be different for you, depending what account you're using. But for us, we're going to be using this one for the demo. And now we just wait for an allocation. Hopefully this won't take too long. Right, we're in. Now to run it, you can just go Slorado basecall like this, and it should tell us everything we need to start a basecalling session. So here it's telling me I need to specify a model. So if you remember from before, we do have a models directory. We can just use the fast accuracy model for now. And I do have a one read 5 kilohertz below 5 in our directory. So if we run that, it'll spit some information back out of this. This first one's going to take a while. It's not usually this slow, but the GPUs do have to wake up on this node. So it should be about like 20 seconds. But okay, so if you see here at the bottom, we have our performance benchmarks. As a user, it's probably not too important for you, but as a developer, this is great for like debugging and benchmarking your solutions. So up here we have our output. This is our read. Now you probably don't want everything spitting into your command line, so we can output that into a file. We'll just call it reads.fastq. And now if we run it, you'll see that it's missing, right? So now if we look into our reads.fastq, there it is, right? So another thing I do want to touch on is performance. So to take full advantage of the hardware you're using, you want to add some options in that Slorado has. So for something like this is Setonix GPU nodes, they offer 64 cores. So to take advantage of that, we can use the -T option to specify our number of threads. We can put 64 there. And then the next option is our GPU batch size. So this is going to determine how much GPU memory Slorado gets to use. This is going to be different depending on the GPU you're running on. So the more memory your GPU has, the larger your GPU batch size can be. And it also depends on the model you're running. So the general rule of thumb is that the more accurate your model is, the less batches you can send to the GPU. But for something like the fast model on Setonics, we can get it up to 2000. So if we run this, you'll see that we have 64 threads, and we're running it on a GPU batch size of 2000. And if we look into our reads.fastq, we have our read there. Now, obviously, you don't want to be babysitting like a 16 million read run all night. So for that, we can use a Slurm script. And to save us some time, we have an example script ready in our GitHub. So we can just go back to the homepage. You'll see that we provide a Pawsey specific guide. If we follow that, you can see we have a example script somewhere here. Let's copy that, paste it in.
Okay, seems like it did.
Yeah. So just to go through a few things. So up top, we have our Slurm variables. We want a GPU partition. Here we're asking for 8 GPUs. That's a bit unnecessary for a single read, so probably just change that to 1. We're asking for 1 node, 1 task. If you're running in array jobs, your n tasks and nodes could be a lot bigger than this. I don't know what the exact limit is, but you can play around with that. So for our below 5, we do have to change this. We can just go to use our single read, and that is located software objects. Demo. Thank you. A fast queue, yep, that's fine. High accuracy is fine. That's also okay. We do have to change our Slorado directory. So same as before. And I think that's everything. So these values are hard-coded. You can obviously pass these in as parameters or environment variables, whatever you want, and customize it that way. And all we're doing is we're hard-coding these parameters to run a basecalling job, and then using srun to launch it. So Control X, save, yep. And now to actually launch it, we use the sbatch command, specify our count. You can do this manually if you remember it, but Pawsey does give you an environment variable that stores it for you automatically. So I believe that's correct. Dash GPU, because we are using GPU hours, and then we're using our example script. So you see we submitted a job to check that it's actually queued up. We run sq-me, and you'll see it's here. And that should be it. Now you know how to base call on AMD. Are there any questions for this specific example?
So do you have any tips for maximizing performance?
Yeah, so I mean, the main 2 parameters you want to modify is the number of threads you're using, so you can take advantage of all the cores that your CPU has. And you also want to maximize the amount of memory you're taking up on the GPU, so you can run basecalling as fast as possible.
Yeah. And to add further, in the Pawsey guide we have provided, in the script there, we have provided for the 3 different models, what are the optimum like GPU batch sizes are. we manually benchmarked and found the maximum size that can accommodate the whole GPU memory. But of course, if you are running on a GPU with a limited amount of memory, you might have to, scale those parameters accordingly. Yeah.
Nice. And I do love that it has the built-in benchmarks as well. That's so useful.
Yeah. So
we did have some questions in the chat. So can it do duplex calling and correction?
Yeah, so I'll answer that question. So at the moment, we only support simplex-based calling. We were at one point thinking about, in the future, developing the duplex calling as well. But ONT, like 2 months ago, they said that they have discontinued duplex calling and you can't buy those flow cells anymore as far as I know. So given it's not going to be something that are in the long term, we thought, no point in us investing a time on it for only those flow cells that existed for a few months. Yeah.
Yeah, that makes sense.
Yeah, and the correction option. So I haven't really looked into whether it is using GPU or CPU only and how the performance is, because in Dorado, if it is CPU only, it can be run on like any CPU, Intel or AMD or whatever, because they provide like x86 binaries, and both AMD CPUs and Intel CPUs are x86 64 architecture. But if it is using the GPU, then yes, it'll have to optimize or rewrite whatever the part which is not open source. But at the same time, it seems like this correction part is only very unstable at the moment. They haven't really made it stable, and we don't yet know where it will lead to. So we believe in the future, if it becomes a very stable feature, we will look into implementing that too. Yeah.
And so is there a minimum requirement for the AMD GPU? Like, I assume this means to do with the version of the hardware. So obviously we have MI250X, which is relatively new.
Yeah. We had access to some other GPUs through the AMD hack cluster, where we tested on MI100, which is the previous generation
MI210.
of the things. And we also checked on MI210 GPUs. And also, I think we tested on MI50 or 60. That's even like a lower end GPU. So we tested on those set. Yeah. So I believe, we haven't had access to any other GPUs to test, but we believe that if they have like 8 GB of memory, at least it would run. Or if anyone needs help in getting on a different GPU, you can always open an issue. We can try to help with any compilation options or something. Yeah.
Okay, that's great. And so from Andrew, so Slorado has only version 4 model support from ONT. What are the barriers to version 5 support? And in general, how do you plan to advance Slorado as ONT releases updates to Dorado and all the models?
Yeah, we've already implemented it for V5. So that supports the transformer models, which I'm guessing is what they're referring to. We're just on the final steps of verification. So we've already done this for AMD, Pawsey, and we're just finishing up on NVIDIA. And once that's done, we're going to release the binaries for that.
Okay.
But if you want, if you're really excited about it, you can go into the dev branch and compile it yourself if you want. It's up to you.
Yes. So at the moment, the development branch of Slorado already implements the support for V5 models, But as Ponson said, we want to base call on a huge sample on multiple architectures with different conditions to make sure things are working as intended and the base calls are not rubbish, but correct base calls for all the cases. So that takes a bit of time. So once that is done, we will create a binary which you can just download like we did now and then extract and run. Yeah, usually more than the development is the testing that takes time because you need to like exhaustively test multiple parameters, multiple types of GPUs and so on. Multiple models as well. So there are many variables to change and test once you do a major like a change. Yeah.
Yeah. And so I also have a question for applying to use a GPU allocation. So on Pawsey, if you don't already have a GPU allocation, you would need to apply for one. Probably the quickest way to get access is through the Biocommons Ables scheme, which is also what Jacinto and Bonson use to do their testing. So if you already have particularly if you already have like an annual allocation, but you want to top up with GPUs, I would definitely apply to Ables to enable you to get access. Otherwise, you would by default just have a CPU allocation. Or if you've not used Pawsey at all, that's also fine. You can still apply through Ables or one of our other schemes. So I think, though, now that we've gotten through those questions in the chat, shall we open the questions to the floor for a proper live Q&A with the audience? So if you'd like to be part of the discussion, as Karina said, feel free to turn on your camera and put up your hand to ask more questions. I think Ash might have had a question earlier. Christina, go ahead.
Yeah, hi guys. It's nice to see you in video finally. We had a few chats, so it's good to see people who develop software and people who use them, so that's great.
Thanks, nice to see you too.
Yeah, nice. Just wanted to give you a little bit of feedback on my experience. And so I need base calling very extensively. And I was considering using Slorado. I want to give it a try still. What I think it's a little bit missing, it's all these tools that need to be developed on Dorado, like the multiplexing and base correction, that will help a lot because we use them in our pipeline. And this as an end user will be greatly appreciated, but I know it's a lot of work. You're compiling, you're testing all the parameters. And yeah, just this, I ended up using Nectar for base calling and for our pipeline. And but I'm keen to test I'm definitely keen to try those on Pawsey. That's on my plans. Just getting a little bit more support for the other tools will be great. Yeah, that's my feedback.
Yeah, I mean, we look into implementing some of the options when we get these V5 models. But one challenge for us is because we are looking for any research opportunities. So just porting another feature to a tool is not really something we can afford to put a lot of time. So that's one of the limiting factors for us. Yeah, and similarly, because these things like demultiplexing and everything, it doesn't really even, I think, use the GPU, right? It just simply use the CPU, I think. So yeah, so that's therefore because there are other ways to do it, we were not really prioritizing those kind of things. But yeah, once we get these major features done, yeah, at one day we might be able to go and look into those kind of things.
Yeah, that being said, Slorado is a very simple and lean framework. So if there's any base caller to make your own modifications or tinker around in, Slorado is great for that. You can go in and maybe even learn how base calling works yourself. It's that easy.
Cool. Have you considered to directly work with Dorado and get advertised through them, getting this to become a mainstream?
I mean, yeah, I mean, that's one of our probably goals one day, like when it is, like relatively stable and demonstrated that it really can be used. Hopefully, like we'll be quite happy if like ONT takes over the thing and integrate to Dorado.
Yeah.
And yeah, I think, then we can switch to another interesting project. Yeah, one of the dreams.
So all our code is open source, so
yeah.
nothing's stopping them from just like copy pasting our code into theirs, and Dorado supports AMD. That's the point of open source, right? I did set up the library in a way where it would be very easy for them. It's like one library call. It's exactly the same as Koi, right?
Thank you.
Ash?
Thank you for the interview and the presentation. It is quite interesting. I'm not coming from the biomedicine background. It's more, I was interested on how your conversion process from CUDA to HIP. So as a point of reference, how long did it take you all to do that conversion?
So in terms of the actual implementation, it was very easy. It was literally copy and pasting the CUDA API calls into like a HIP file, and then changing, using like a macro, like changing the word CUDA into HIP, right? There's only like a couple very small things that you have to keep an eye out for. So some of the kernel code, you have to know like how big your warp size is. So for AMD, your warp size is like 64, CUDA is 32. So you're gonna have to change a little bit in that. And then another thing is that some functions in CUDA aren't supported in HIP. But there are like very similar kernel codes that you can use, which is what we did. So in terms of like implementation, it's very easy. The hard bit was compiling it, right? Maybe you want to give some insight into actually compiling for.
Yeah, because we had some challenges like trying to find out the correct library versions for ROCM and this things. Yeah, But before that, I think like because Koi was not open source, Bonson had to look at the CPU implementation of this beam search algorithm and the decode algorithm and convert it logically to be suitable for a GPU, because the code that runs on a CPU is not directly suitable for the GPU. You have to restructure the algorithm to use all the parallel like cores. So that, rewriting of the algorithm in CUDA was, I think, one of the like time-consuming ones. But once you do that, porting to HIP is very straightforward. Yeah, and after that, compiling became a bit of a challenge.
Yeah, I mean, I completely understand the process, because that's exactly what we're doing. We're refactoring everything into CPU code, making sure it works, and then slowly migrating back onto AMD GPU. So it's just taken some time. That's all. But yeah, thank you very much. I will definitely look into your source code as well for some hints, which we can also hopefully follow on.
Yeah, so on that, you might want to look at the OpenFish library. So you'll see as a submodule inside Slorado. And OpenFish is like a way smaller, cleaner project that you can explore like compiling options like what we did. So that should be a lot more helpful than digging around in Slorado.
Okay. So is that the one on AUS Ocean /OpenFish? Is that the one?
Yeah. It is the submodule inside the Slorado.
Yeah. Okay. Okay, cool. Thank you.
Because then we don't have the complexity of like integrating with Torch and other libraries for OpenFish. It's just a self-contained project where it takes some API calls and input data in binary form and do the GPU calls. Yeah, so that's relatively easier to compile, just works. But it's that, linking all the libraries together was the hard part. Yeah.
Cool. Thank you.
Does anyone else have a question? Yes, Henry.
Good day. Yes, I was just thinking, what is your impression of the, accuracy or reliability of the algorithms in general? Like, if you have, what would you think is the error rate of these algorithms in general? Because in the lab I'm working for, we have both like, new samples from like modern, but also like very old samples that may have contaminants and things. And I think there are, other kinds of, what to say, with the GPUs, they can be both single precision and double precision. So what do you think affects the accuracy and the reliability of the nanopore technology?
Yeah, so I think Bonson, can you open the blog article where we have the
So are you asking for the precision of nanopore basecalling in general, or Slorado versus Dorado?
Yeah, I guess more in general, but I guess it would be interesting to know what comparing, different GPUs, what kind of accuracy you can expect from different kind of GPUs, both AMD and NVIDIA, and maybe in general as well, but it's.
Yeah, so I mean, we have seen like, let's take like Dorado from ONT, and when you run it on two different like GPU architectures, you get slightly different base calls.
All right.
Very slightly different base calls. The reason is the types of floating point precision and stuff on the like different architectures. But I mean, we have checked that there are pretty minor differences that wouldn't really translate into any other like major changes. When you do variant calling, for instance, that effect is gone all the time. And also most of these little changes come from bases where the quality score is anyway low. So I believe the effect due to the precision is not that like significant. Yeah, so here you could see we use this thing called the identity score to measure the accuracy. What we do is after base calling, we align them back, the reads back to the genome using Minimap, and then our screen is not shared.
Oh, is it not?
Yeah.
I should be.
I think it's shared, right? I can see it.
I can see it.
Or is the graph figure 1 there, or I guess?
Oh, yeah.
It's mine that it was pop out yet.
Okay. Yeah. So we use Minimap to align to the genome, and then we calculate this thing called the like BLAST like identity score, which basically tells like a value from 0 to 1. If 1 means it perfectly aligns with no changes in the bases. So you could see like the Slorado with AMD and NVIDIA, and then Dorado, original Dorado with NVIDIA, which is orange color. Slorado is blue color. You could see that in some cases, it was even slightly improved accuracy. The reason we believe is Dorado uses 16.
They use it's a quantization for the model. So they do in Dorado, they sacrifice some accuracy for performance. We haven't done that optimization yet in Slorado, so that's why you see us like edging out a little bit in accuracy.
Yeah, we don't exactly know, but that's what we believe is the reason. So, but as you can see, it's a very like minor like difference that you wouldn't even see after you variant call or any other downstream analysis. I hope I gave some insights into your question.
Yes, that's interesting. I was just wondering also, like with these data sets, how large can they be, or like hundreds of gigabytes or even terabytes? I don't know, these kind of sequencing at most, because I know it can be a bottleneck when you have very large files when it's converted to 4K, and try to use Dask to analyze, FASTQ files in after the base calling has been done. But so it's, I don't know what what's your experience with the large files and how to better better treat very large files with the output? Is there any kind of strategy to employ to better because that's I guess what GPUs is very useful because you can treat much more data, but I guess that can be a bottleneck even if if the data is very large, So,
yeah, so I think, for example, this particular data set we used, it has 20 million signal reads, and that was close to, I think, 750 GB of BLO5 file, the input. Yeah, but what found was, because in Slorado, when you run the thing, it prints out like which step took how much time, like IO took this much time and the inference took this much time. I think the IO was not even close to being a bottleneck.
Yeah.
Yeah. This was a big problem in this ONT's previous fast 5 format, that's how, our slow 5 format also came. It was so slow and annoying to work with. For a joke, we made a format called slow 5, and then it got real. But with that kind of file formats, if it is properly engineered, then IO is not a bottleneck, especially on places like Pawsey, where the file system is, I think, cluster-based, which is a very high-performance file system. It is really good if you are streaming. But if you start doing random access, it's going to be not fast. But Slorado here just sequentially read the file. Therefore, it's not a big bottleneck here.
Nice, very interesting. Thanks for the talk and presentation. I don't know if I had more questions, but so anyone else perhaps have more? Yes, thank you. That's good.
Excellent question.
Just a comment. It's great that IO is not a problem for this, but if IO was a problem, Pawsey now has NVMe, which is like near node kind of memory or storage on the GPU. So there was a use case actually that came to my attention yesterday, someone doing this big training using PyTorch. And so they're getting a big slowdown on Scratch, but we were able to move them onto NVMe, and they had like a 1.8 times speed up, which was pretty great. So that's also an option if you are finding that you're IO bound on, on those type of tasks. So something to think about. We're going to, I think, need to write more about it because I don't think we've advertised it enough. So watch this space. Keen for more questions though, if anyone else has got some more. Otherwise, we can.
Hey.
Oh yeah. Is that a question?
Yeah. I'm wondering if Slorado supports methylation basecalling.
At the moment, we haven't implemented the methylation calling yet. Yeah, so we asked after I think the V5 model implementation, we will have a look into how difficult it is to implement it. Yeah.
Okay, thank you.
And yeah, also on the top of that, also like RNA sequencing, it's another thing to look at, like the models for the...
Because at the moment, this only supports DNA. We haven't gone into, because we wanted to start it with the thing that is most used, So it's like, and the largest data comes from DNA. And mostly use case is doing simplex based calling. So that's why we started with that. I mean, there's a lot of things to do in the future. We will slowly work towards that.
Henry?
Yes, I was just one with Sarah's comment in case if that's a general feature of GPUs, you think with the extra memory bandwidth or what to call it, if it's general feature of the NVIDIA or AMD GPUs, or is it a special feature you have at Pawsey HPC, the memory, extra memory for?
It's not everywhere. So we only had our NVMEs put in fairly recently. So it depends on the system that you're using. So you might find that a lot of places do have them, but it's not guaranteed. It is pretty good though. Like when you see the performance out of it, it's pretty nice.
Oh yes, I know there's like a maximum limit of certain GPUs, what kind of data they can handle, but yeah, I think those kind of things will be important as data grows, and so it's indeed, and they even use like Dask Python one can use for handling a large amounts of data, but it's one have to get it to work on different systems, and so it's oh yes, it's good to discuss those, how to solve those kind of problems, but with large data. But it's so all right, that was my only comment for that. So thanks.
Any more questions? Now is definitely the time to ask. Otherwise, we'll move on to wrapping up, I think. Next step.
Yeah. Hi. I'm curious to know if you'd be open to like some collaborative projects on improving Slorado.
Yeah, definitely.
Definitely.
Let's look it up for you. Can feel free to open the issue. Shoot a message.
Yeah, okay, this room it's got too many fancy features as you can see, and it somehow turns the light off if you don't see a movement. Anyway. Yes, yeah, so we are pretty open to collaboration, and that's, I think, the goal of an open source project like this, where people work together to provide, like support and time for implementing various features.
Yeah, if you want to implement methylation-based calling for us, feel free to do that as well. We'll definitely merge it into our branch.
Sure, thank you.
Awesome. I mean, yeah, there's definitely, I think, a huge amount of work to be done in this type of space. So the more hands, the better. So more questions, comments, people volunteering to work with Jacinto and Bonson. Going once, going twice. All right, closed. So let's move to closing remarks. Jacinto and Bonson, what's the next step for interested parties that want to get stuck into Slorado?
Okay, I think the next steps would be like following what Bonson like showed today, and first you can get started with a little example data set. If you go into the page on the repository where Bonson showed like the Pawsey page. Yeah, there we even provide a little like 20K data set that you can just download and run. So just follow these instructions and see whether you can get it running on Pawsey. And then try to do it on a data set of your own, probably with a smaller data set, and slowly increase it to a huge one and see how it goes. And you can also read the blog article if you are a person interested in technical details, like how we implemented the open source part, HIP-related stuff. Also in the documentation, you can find any like things on parameters and stuff that you can see into fine tuning. And you can always like open a GitHub issue if something is not clear or if the documentation should be improved. Yeah, any suggestions, like, for example, like if we get a lot of suggestions that please implement this feature, yes, then something we can look into. yeah, like that.
Right.
Yeah, we have to like if implementing a feature, if it is something that is going to be widely used, yes, then it's definitely something we are happy to invest our time and implement. Yeah, yeah.
Awesome.
So
And
I
I have to
think
say, the documentation is really nice and clean. Like it was really good. And if people want to get onto Pawsey, I did put on in the chat, sorry, the ABLE's bioinformatics specific access scheme. So that's a really quick turnaround, anything from really small allocations to quite large ones. So Biocommons has done a great job with getting people access that way. And also, if you are using Slorado on Pawsey and you're having some Pawsey specific issue, we do have a team to help with that as well. So you would lodge a ticket through our help desk, and then a supercomputing applications person would help you with that if they need to.
yeah. And if any one of you have any other like systems with like different GPUs, feel free to try, and like if it doesn't work, open an issue so that we can look into what's causing it and fix it, because we tested on all the possible things we could access, but I know there are like many architectures out there. So try and give us like the log or something if it doesn't turn out to work. Yeah.
And that's awesome that you're so open to responding to issues and improving the features as well. Like that's great.
Yeah.
Okay, well, if no one has any further questions or comments, I think we can thank Hasingdu and Bonson, and as well as Karina and Aditi for helping put together this event. It's been really great to work with you and to learn a lot more about what you're doing, and I'm really excited for people being able to use this. So thank you so much.